Right. So a lot of what, as Christian was alluding to, that I've been really obsessed with for a long time is kind of, nicely outlined here, where these A, T's, G's, and C's are the nucleotides that, if you put them back into the embryo, will actually give rise right here. So this is the classic Eve stripe, where they have these beautiful stripes that'll reproducibly come up, and you have stripe 2, 3, and 7, and those are actually driven by these nucleotides here. So a lot of what we've been, really fixated on in the group for a long time is understanding, mechanistically, how does this really happen? And then a lot of the things we're continuing to explore is not just this one little enhancer, but rather, as I'm showing the adult here now, because we're interested in the whole networks that kind of lead to this body patterning and ultimately, this level of selection that operates across these different spheres. So I always like to kind of start off with like what I think is the major challenge in the field. Obviously, this is massive and something that I'm not going to solve alone, but I think it kind of puts a lot of what we're trying to do into context, I think, which is that, the thing that we can do very, very well right now as a community is this little genotyping bit right here, right? So we can go out into the wild, we can grab any single fly right now, we can grind it up and sequence it. I know many of you are very, very good at doing that. But the thing we're really struggling now as a community again is connecting these genotypes in a meaningful way with a lot of things that I'm interested in understanding, which are the phenotypes, right? What I mean by that very specifically are things like this right here, what I call internal phenotypes, right? So you can have, what I'm actually showing you here is a slightly later embryo. So this would be the developing head and the tail, and this is belly, which is facing up. And again, you get these beautiful stripes, which is actually governed by gene expression here. But there's a huge number of other things that you could classify here, such as metabolism, cells and tissues, how they manifest to external phenotypes, cross development, giving rise to different morphological differences, behavioral differences. And ultimately, what I'm going to kind of tip back towards, which is a big push within the group, and I think many of you guys are quite clearly interested in this here, which is the environmental interplay, right? So the reason why I'm still fixated in using the fly as a system here is because really what you can do now is, create very custom genotypes, which is a lot of what people are doing in the group, and then really see over time how these will manifest across different populations, right? So again, this environmental bit is something that's really new, and people in the community are really kind of scratching the surface, I would say, for this. And a lot of what I'm going to focus on through the talk today is first, looking at kind of my classical work, looking at internal phenotypes, in this case, in gene expression patterns. And towards the end, I'm going to tip into something that's quite new for us, but I'm very, very excited about this potential. And in particular, I'd love to keep discussing things from more the PopGen perspective going forward. So that'll actually be in the kind of environmental interplay across these different networks. So a lot of the work which I'm going to focus on at the first part of the talk then is this little sphere right here, which is these very classical enhancers, which I'll walk you through really what they are, but you can see that they actually bind transcription factors, which if you put in front of reporter genes, so it could be GFP, DS-RED, anything of interest, if you put that back in the fly, they'll actually give rise, again, to these very nice patterns of gene expression. So this one axis here has been, again, a lot of what I'll be first focusing on, and mechanistically what we've been diving into very deeply within the group. And why are we interested in understanding these particular elements? Just to kind of bring everybody up to the same baseline, if you haven't been studying enhancers obsessively for a long time like myself, is that we know very simply what enhancers do is, well, maybe not simple, as I'll walk you through, but enhancers will actually turn genes on, right? So you can think about them as little switches that are turn things off or on, different parts of development. So you could have enhancer A, for instance, which will bind different combinations of transcription factors, which will turn on that gene body here. And you could have a different enhancer, which will be located somewhere else, either further upstream in this case, which will bind different transcription factors and in turn that on different tissue types. So what we mean again by that is you can see, for instance, this one might turn it on developing limbs if you're, in this case, for a vertebrate, or it could be within an embryo, a nice stripe. And again, this other one, which could turn as enhancer B, might turn on in different tissue types, right? So this is, again, these switches during development. And one thing is that's very important from an evolutionary perspective, you always kind of seed in the back of your brain, is that there's far more enhancers in coding regions, right? So it presents a very large target then from an evolutionary standpoint. Okay, so again, why do we care? And there's many, many, many examples for how enhancers can drive and shape evolution and as well as disease. And I'm going to go show very classic examples here, which is brain evolution right? So a lot of the prefrontal cortex we know is driven through new enhancers. We have things like placental, entirely new tissue types. We have pigmentation, so again, getting a little closer to home. For this is actually the developing wings, or actually not developing, in this case, the adult wings of these fruit flies, right? So we know that a lot of these differences here in pigmentation are governed by changes in regulatory regions. And we have other classic examples like the work coming out of the Kingsley groups, and many other people have coming through these different groups here of the armor plates and sticklebacks. You know, you can go going on and on for many examples of these. There's limb evolution, for example, and these same regulatory regions we know also will manifest towards differences in humans, which might arise in this case through, you know, different changes in the patterning of your limbs, right? So again, there's many, many different examples of these, but what I want to kind of emphasize is oftentimes they're very like these case studies. So a lot of what we like to do is go very, very deep, and I'm going to walk you through some examples to really understand basic principles of ultimately, again, what we're trying to do is put this in a more predictive framework, which I'll touch on in a little bit. And again, from a disease perspective, we know that about 90% of human genes are resulting from differences or changes or SNPs within these particular coding or non-coding regions, right? So if you play with one of these switches, it can have very strong effects. In this case, like I'm showing you in this extra finger here for this human. Okay, so how do enhancers do this? And this is we're going to kind of do this deep dive into really focusing on this here. And this is a little piece of DNA that I've been fixating on, again, for a long time. And the reason is that you have these very simple, again, letters here. If you put these in front of a reporter gene and put it back into the fly, right, this 300 base pair piece of DNA, we know that very reproducibly, you'll get these nice, I think, beautiful stripes of expression, again, in the developing belly of the embryo, right? So you get these very nice stripes that will manifest over and over again, right? So what we're trying to understand then is how these enhancers, right? Because again, what I'm showing you here is kind of this textbook view where you have these very simple assemblage of these little elements here, kind of this cartoon form that will turn this on. But what I kind of want to highlight here from a mechanistic standpoint is that we struggle to really predict these enhancers. What I mean is if I just gave you a DNA sequence of an entire genome, there's really nobody yet who can go through there, but we're getting much better at it to really pull out and say this would be an enhancer. And what's even more of a difference, a struggle then, is to predict even then where it would turn on a developing embryo and the effect of mutations on this, right? So clearly these are very important things to study. The question is, why do we not understand this? So this is something we've been studying quite heavily again, and to understand what information we're actually missing. So one of the approaches we did then for this is to understand, you know, very simple questions like how many binding sites might there be? How robust are enhancers if we start to mutate these? How evolvable are these enhancers? And then we could put them ultimately in a predictive context, right? So these are things we're trying to go towards as a group again. And the reason I'm so fixated on this one piece of DNA is for me, it actually integrates an enormous amount of developmental biology that's been done. And in this case, a lot of this work was done originally by Yanni and Eric, shown up here, in what is known as now as the Heidelberg screen, which ended up winning them the Nobel Prize. And the reason it won the Nobel Prize is because, as you can see here, which I'll walk you through in a second, is that this screen actually identified almost every single regulatory network that is conserved from flies to humans, right? So these are Hox genes, you have EGF genes, hedgehog, notch signaling. And what's important for a lot of what I'll be talking today is you can see that a lot of these are converging on this little gene known as shaven baby. And the reason it has kind of this ridiculous fly name is that shaven baby is actually the gene which will give rise to these little hook-like structures, which is what Yanni and Eric are actually using in their screens. So what you see here, this is actually the larval, so an L3 larva. And you can see, if you've seen David's talk, I guess he was here last year, you can see these nice denticle bands. And so what these denticle bands are assembled with, these are, if you zoom in, are actually little hooks again. So if you don't have this gene known as shaven baby, you do not have those hooks, and it's totally lethal. They can't crawl, they can't eat. So again, what this little piece of DNA, though, the reason we're so interested in it is you see these 8 little bands here. These 8 bands will actually correspond to these exact same cells as you can see right here. So what we have then is this whole history of many, many groups now who have worked on unraveling the different kind of genetic and developmental networks here. Many of them are converging on a little piece of DNA like this, which can kind of function as a readout for us, right? So now we can start to understand, as we mutate this particular enhancer, the direct effects on this. And really to kind of highlight this again, what these particular regions are doing, this is actually a crawling larvae, right? So now you can see that these little lit up bands are these similar denticle bands, right? So to really put this in context, right, this is what these genes are controlling to allow them to kind of crawl through their food source. Okay, so now again, we have this kind of classical view of the cartoon view of an enhancer. And so what we want to do is to step away from that. And what I want to highlight here, kind of the challenge in the field, is that this is kind of, if you go through and pull out different flies globally, what you can see now, if you go into one of these regulatory regions and you do all the alignments, is that it's actually very sparse amounts of variation for us to actually capture to understand really at scale mechanistically how the impact of an individual mutation on this expression pattern. Right? So there might be some limited variation in these natural populations. And what's a kind of a challenge for us is that we know if we start mutating some of these now, this will have very deleterious effects on these systems, right? So now we're kind of, we have all this contingency, drift, which many of you guys are struggling with, epistasis, and fitness as well, right? So basically what we have now is kind of a limited view of what's fully possible, right? So this is what we wanted to explore in a much deeper way. So the way we kind of went after this is much more of a way from a lot of these more synthetic biology backgrounds. What we did is we actually took an entire library and we took this enhancer and we actually just sprinkled it with around 2% mutation rate in a totally unbiased way. And then what we did to actually assay these is we put all of these in the exact same spot in the genome. And what I want to emphasize again is that these are reporter genes in the same spot, but by doing reporter genes, what it allowed us again to kind of decouple the effects of a mutation now from what might be happening from a very deleterious mutation. And a lot of what we've been doing then to get the throughput up, because 1,000 lines is a bit of a challenge here. And so we spent a lot of time building a lot of fixations and staining systems to fully automate this whole process. And on top of that, we spent a lot of time now to fully automate all of the ability to scan all these embryos in, right? So what we wanted here was not just to be lazy, but was also that this brought us an enormous amount of accuracy and precision in a lot of our measurements, right? Because you can just kind of standardize everything you do. And now to put this in a much broader context for a lot of what we did, and I kind of like to put these slides in particular for PopGen audiences, is that now what I'm showing you from that library now, it represents the same spot in the genome. There's a huge amount of variation, right? So now this kind of explodes out the space, and this represents just millions of years if you compare this to just standard mutation rates. Again, all at this one piece of DNA at the same spot in the genome. So it allows us to really go deep then. And again, because now this is not constrained by anything else, it's to really understand this input-output relationship of how this enhancer is working. And one of the major things we found here is, so I'm going to show you kind of the psychotic video, which goes very, very fast, but this is kind of by design because we found what I think is a bit of a jarring thing. So this is actually the wild type enhancer right here. So you see, again, this beautiful expression pattern in the fly. And now as I play this video, every single frame now is actually going to be an example of a different line from those 1,000+ lines or so we sequenced. Let's see if I can get that to work. Okay, so you guys see that now? Yeah, see? So what I hope you can see is that there's an enormous amount of variation from these lines, right? So these are not the same thing. I kind of went into this assuming this would be a very robust enhancer, and instead, as you see here, you've got kind of a full gamut of different expression patterns, right? So to kind of stop this and slow it down a bit, this is the major thing that we found through a lot of this results. What I'm showing you right now, in this little corner right here, this is actually the wild type enhancer. Each one of these is a single mutation somewhere in that enhancer. And the major thing that we actually found, and you can see all this quantitatively here, and again, I really have to thank Timothy Fuqua, who's my first graduate student who really pushed this project, because what you can see is that statistically, every single one of these different mutations had an effect on expression, which for us at the time was very, very jarring and totally unexpected. But the nice thing about having the scale of these libraries, what this allowed us to start to do, and I won't go into scale about this because some of this is published again, is that it allowed us to start to these, like we call little footprint scores, where now if you compare across these libraries, you can start to see the amount of the impact of if you mutate in this region, the probability you're going to really collapse expression. So you can start to see that there's a huge amount of expression or important information scattered around this enhancer. So again, all mutations changed expressions and timings, levels of location throughout the embryo. And what, again, we concluded here is that these are very, very dense, right? Meaning that from a mutational perspective, as you mutate anything here, we are getting an impact on expression. So again, it's nothing like this textbook view, right? So this is kind of, again, what I kind of almost assumed going into this. Rather, it probably looks something more like this, right? So these are all known and vetted in the color ones, binding sites for different transcription factors. And many of these, we don't really know what might be binding there in these little gray sites. It might be structural or something that's not even related to a transcription factor. But again, from the information perspective, these are very, very dense. Right, so just kind of highlight that again here, information dense enhancers. What we are curious now from an evolutionary standpoint is the impact does it actually have, in terms of constraints or other things across the system, right? So we did not expect them to be this sensitive going in. So what about other tissues or different enhancers, right? Is this kind of a general thing or just something weird for this one element we are exploring here? So we actually went through and started to explore this from the constraint perspective. And the reason we did this was, I'll kind of walk you through right here, because again, these are that same shaving baby enhancer. There's that wild type now, and I'm showing it in green. And each one of these is actually a different mutation, right? So again, you can see that they have slightly different patterns of expression. And then what we started to do is look across the different tissue types. So I'm showing you here is actually ectodermal tissue as it goes higher, mesodermal regions in the embryo, or endodermal regions. And the thing that was quite jarring for us is, again, you see these different patterns of expression in your native zones, but you get no expression outside of this, right? So it's for me, which I did not expect, right? To really, really emphasize this. All of the variation we were seeing was in native zones of shaven baby expression, right? So some of these might be a bit precocious because we have these interesting ones like this right here. This would actually be in developing wings. But again, in the stage later, this is where shaving baby can start turning on there. And again, these other ones, you start getting new patterns of expression, but these, again, are the same tissue types that shaving babies normally expressed, right? So for us, this is actually very, very interesting. So we are curious, again, to take this even further and see how this might shape other tissue types or enhancers. And the reason we're interested in this is kind of this classic Waddington metaphor, right? So don't dig into it too much, but I'm kind of using this to emphasize a point here, is that shaven baby, that original gene I've been walking you through, is the definition of a terminal selector gene, meaning that if you express very late in development, if you express shaven baby, it'll actually, again, acting as a transcription factor, will turn those cells into a trichome. But what that effectively does is kill that cell type, is you're basically like cuticle or like fingernail type material. So we were curious if other ones, maybe if you go slightly earlier in development, such as this gene here, or even at the very, very early specification of tissue types, maybe these are a bit more plastic, right? So they might evolve slightly differently, or as we start mutating them, they might give slightly different patterns of expression. So Noah in the group jumped on this as a new post or graduate student, at the time actually a master's student, and she took another very classic enhancer known as Tin Man, which again is not that important, but the important thing is that it's expressed throughout the mesoderm in kind of these mid-stage embryos. She did the same thing where we kind of took this mutational library of that enhancer and just an unbiased way sampled the effects of the expression. And very strikingly, much like that shaving baby enhancer I showed you there, now if you start assaying the effects of expression, again, we can actually get up or down slightly here, unlike maybe shaven baby, but we never get any expression outside these zones, right? You can see that you just get nothing here as you start assaying across many, many different lines and different regions of the ectoderm or endoderm, right? So again, super constrained. And then we went even earlier in development. So again, these enhancers aren't necessarily that important in the names. What's important here, though, is that this particular one, known as twist, will turn on and specify the early mesoderm. And this is really early. At this point, if you were to take these embryos and cut them in half, showing you here, these are basically balls of cells at this point. So this is really laying all the ground plan for the early development. And here's another enhancer I'm going to compare it to, because this is one that's known as rhomboid, which is, again, isn't that important, but basically what's nice here is you get this nice stripe pattern that would actually go down both sides of the embryos, and this would actually lay the groundwork for what will end up being the nervous system. The reason we focused on these are very classic again, coming out of Mike Levine's and other groups, and I'd spend a lot of time kind of mapping a lot of these binding sites and others. So what we did is now to look at these, again, different tissue types, the ectoderm, mesoderm, and endoderm, to see, again, how constrained these might be. And in this case, just like I showed you before, as you start to mutate twist, now as you mutate it is, again, only in the mesoderm, you're really not turning on any other tissue types. And again, with rhomboid, the same thing, right? So we see these similar patterns emerging now over and over again, where you're not just turning on willy-nilly all across the embryo. We actually looked at different stages, which I'm not going to show you right now. But the important point is that all change in expression patterns were in native tissues, right? So for us, this is quite interesting. So we're kind of suggesting right now that existing elements are far more functionally constrained than I think was really appreciated previously, right? Because I kind of thought going into this that these are easily tinker with these, and you're turning on different tissues, and it's going to just facilitate all sorts of evolution, which we didn't see here. So then we became interested in this case about de novo evolution, right? A different way to kind of escape this was if you can't tinker with something that's kind of broken outside of kind of level effects, maybe you can just evolve something new. So how easy is this to do? So then we actually went through the same pipeline as I showed you previously. The difference now is instead of breaking enhancer, now we're actually taking totally random DNA, right? So this DNA that has never seen selection before, right? So we're taking random bits of DNA now, doing the same exact thing in front of a reporter gene. And the difference here, which was still really kind of compelling when we first get this data, is that in this case, extensive expression we found from these. In this case, about 36% of these totally random bits of DNA were driving expression across these embryos, right? So you can see many of these right here, right? So you get to see some of these nice stripes. You see different ones turning on, for instance, in the gut, different regions of the ectoderm, right? So you get all sorts of different patterns of expression in a very, very simple way, just through random DNA. But then going into this, we wanted to see what this could actually teach us and inform us about kind of, again, the mechanism for how you might be driving these patterns of expression. So what we did here, and this is actually the work for, I should highlight, this is the chief postdoc on this, is a technician in the group, and Gilberto was the one who actually went through and actually started assaying to understand from a motif perspective, if we look at these, what this could actually teach us. And what he found is that very simple signatures would pop out, because now if we have random DNA that's expressed nicely in the developing gut here, every single one of these, 100% of them actually had this GATA motif. And we know this is a transcription factor that's expressed there. And similarly, if we started looking at this ectodermal, we had these nice stripe patterns. We had another motif that would pop out, in this case, one known as grainy head. All right, so key motifs we found were associated with these developmental expression. And what we predicted then kind of going into this is that one way you could drive this is that these two transcription factors are actually part of a larger family of factors known as pioneer factors. Because what they will do is you actually have this very nucleosomal DNA. The thought is that they'll actually go into those, and what pioneer factors can do, if you have this type of activity, it'll actually open up that DNA, making it accessible so other things can bind and actually regulate expression. Right? So we were curious then is that these pioneer factors, that's what's driving a lot of this evolution or these de novo elements that we're putting in. So we actually bias this library to the same thing now, but by actually spiking in these key motifs. And what I mean by that is right now is this a very classic pioneer factor in Drosophila. So again, it's not really important, the names, but this one known as Zelda is expressed in the early embryo and also in the nervous system. So again, we made the same type of library approaches, but now we have the motif in the middle. And what happens now is if you actually do have this motif with random DNA flanking it, now we have almost 88% of these were driving expression. And what was, I still think, quite compelling about this is that many of these now were in particular zones where we know Zelda is expressed, right? So it seems like it's very internally consistent. And it's just to kind of emphasize, these are not low levels of expression. So now if you actually compare these across these different tissue types to this E3N enhancer that I was walking through earlier, you can see that many of these are actually very, very high levels, right? So it's actually, for us, much easier to get expression than I ever anticipated like this. And this is another beautiful one, because in this case, this is something that's very, very rare for me when I do these type of experiments, where now if you have grainy head here in the middle, right? So again, you're biasing these libraries. We had 100% of these now are driving expression, right? So again, they're not always entirely consistent, but what you can see is that very nicely, many of these zones of expression now are where that factor itself is expressed, right? So we really think that these is a way to facilitate a lot of the expression we're seeing throughout these different systems. And now to take it even a step further, what we wanted to do is to see, what is the regulatory potential if we go into these genomes and look for actual motifs that now might have evolved de novo in one lineage and not the others. And what I mean by that is that these are just kind of a collection of different regions in the genome where another transcription factor, which I've spent a lot of time looking at, we know binds very nicely these regions known as, this is a Hox gene, so again, not important, but you can see you get these nice regions of binding throughout the genome. And here's a nice example here, and you can see that this is, again, a de novo motif, right? So it's come up in that particular lineage. We know it's binding an element, and now when we test these in vivo, you can see you get these very, very beautiful expression patterns. And we think that these are functional binding sites potentially, because now if you mutate these, you actually lose large swaths of that expression, right? So again, we think that this is just demonstrating that these do have a lot of this regulatory potential now that evolution could kind of tap into, right? So to kind of walk you through this first bit of the talk, again, we think that existing elements are functionally constrained. And I started off with kind of these very elegant case studies that have previously been done. So now if you project our data back on top of that, what this is showing us that these are actually examples right here where, for instance, the boa, which lost its limbs, and the loss of armor plates, which repeatedly happens, this is actually the loss of the functionality of enhancer, very similar to what we're seeing in ours, right? So you can lose these very easily. And again, it's something you can actually capture from the phenotypical perspective. Other ones like these two right here, a lot of brain evolution is actually driven through totally de novo sites, as I think I alluded to earlier. And we know other tissues that are entirely novel, in this case, like to eutherians, the placental tissue, we know this is oftentimes driven through these huge amounts of transposons, right? So again, these are different ways that you can actually get new material that's driving this expression. So you kind of escape what I think is a lot of the constraint from standing, elements across these genomes. Right? So again, to kind of put this back in context again, we think that, by doing these type of large scale experiments, it can kind of start to teach us these very general principles of how these elements might function. Right? So a lot of through automation, we've been doing lots of other enhancers, promoters, a lot of other work is soon be coming through the pipeline. But what I want to emphasize now as I pivot towards the end bit of my talk, how much time do I have? I'm fine. Is that, while these are cute, elegant experiments, I think you can get access to, get some of these cute publications. For me, what's really, really not that compelling is that it does not allow us to connect in any way to external phenotypes or anything that might be happening in the environment, right? Because again, these are very stripped down assays that can teach you a lot, but a lot of the pivot in the group right now is to explore this much broader space, again, to connect across these scales. And what I want to kind of walk you through what we're doing right now in this bigger approach, and I'm going to show you just kind of one example of this is a lot of the phenomics thing we've been doing in the group. So we think that by doing these large scale, very high dimensional data sets, this is going to be a way, as you kind of collate these, to really make sense across these scales. Right, so a lot of what we're doing in the group, and I don't have time to go into this, so some of these, if anybody wants to reach out to me after, please get in touch, because we're doing different very discrete synthetic manipulations where we can do individual elements, engineered elements, hypermutate whole pathways. And then as you start to do this, to connect it again using all these multi-omic approaches now to really see what the system can really kind of teach you as you do these perturbations. And again, we're starting to tip into, which is a big reason it's fun to visit here, is kind of these different laboratory evolution experiments to really kind of capture into different environmental changes. So in our group, we're actually calling it developmental phenomics, but many groups have been doing this for a long time, and a lot of the plant people are kind of the leaders of this field. And what I'm going to walk you through right now is, I think, a very compelling example of what you can start to do with this. And again, this is work that's very, very, I think, important, because a lot of what we're starting to understand now is something that I think is happening globally here. And if you like insects, like I do very, very much, this is, I think, a very compelling example. What I'm showing you here is a field site in Costa Rica in 1984. Again, you get beautiful biodiversity of insects. 2007, you fast forward a little bit, again, so a lot of nice insects here. And then you see just a few years later in 2019 where there's this very, very, very dramatic collapse of these, right? So what we want to kind of emphasize right now is that this is not something that's happening local, right? This is really happening across Germany, 2% drop globally every year. Some people put these in different, might be lower estimates. So a lot of what we're trying to understand now from a kind of ecological perspective is what's the mechanistic basis of this right now. And two major drivers have been proposed for this. One is agricultural toxins, and the second is temperature. So these are two things I'm going to walk you through right now to actually understand what might be happening here. And the system we're using right now in the lab, and this is what we're doing a lot of moving forward in the group, which is why I'm talking about it right now, is kind of this new pipeline we've been developing, which now, instead of looking at adults like right here, we're actually taking their L3 larvae, and we're doing these large-scale pesticide libraries. These are very, very controlled ways we can do this liquid media, right? So the reason we're doing larvae, again, is we can completely submerge them and do whatever we need to do in a very controlled delivery. And then in the back end, what we can actually do now, and you'll see why in a little bit, why we're very into looking at behavioral videos, is that this is an example, again, we can look at population scale data, right? So we're not looking at single or smaller amounts of adults. Now we can look at tens or even hundreds, or you can kind of scale up as far as you want to understand the dynamics of the behavioral space for these. So what I'm going to walk you through right now is an example of this toxin screen we did. But really what we did is to understand agriculturally what might be happening and the impact of these chemicals that are being used just pervasively everywhere, is we took everything basically from the Sigma library that had anything to do with any aspect of agriculture. So it's about almost 1,100 compounds. We did 3 different doses, and the lower 2 are very ecologically relevant. This is why we chose these, at 2 and 20 micromolar for many of these. And then again, we looked at the behavioral shifts. And this is going to emphasize why we're so interested in the behavior, is now we're going to show you how we can quantify these in a very automated fashion, because this is an example of just one of these wandering larvae from an example of the video above. And as you'll see, it's going to change color in a second. Right? So a lot of what we're doing this now is we have all these machine learning algorithms that can actually do all of this for us, right? So now we can take whole populations of these, and now we can pull out all these behavioral modalities to really understand, how much time it might be spending in one, how these are varying, transitions between these. And then so now to understand what the impact of the whole library was, right? So it's, it's a bit vague if you look at some of these right here. Meaning that some of these are kind of random names that came out from large-scale chemical screens, if you're an insecticide or herbicide, and you'll see why in a second why I think some of this is a bit vague. So the first thing we actually did in the full library now was, this is the work that's really spearheaded by a very talented postdoc, Lautaro here, and this is the highest dose of toxins, right? So this is 200 micromolar, and what you can see now is if you compare acute lethality to long-term lethality, is that many of these will kill them outright. You can see this 100%, and most of these are these insecticides shown here in blue. But many of these, even at these lower doses, you can see are not fully lethal, right? So these are already in this sublethal regime, and there's a long history of people proposing that the sublethal effects could be very, very critical for seeing kind of these collapse of these systems. And to kind of make more sense of this data, what I'm showing you here, and don't get hung up on the details, but really what this is just a mapping of the chemical space with a UMAP embedding. So basically this pushes apart different chemical identities of these families to kind of make it slightly easier for us to digest the data, right? And to show an example of this, I think, I do have it in this talk. This is just showing a very classic insecticide, which is largely banned now. But you can find other ones that will also co-cluster in the same family up here of the organochlorines, right? So endosulfan, which is still unfortunately used in many places, right? So again, these nicely, maps some of these very classic insecticides, again shown here in these different blue regions. So the first thing we did now is to go very, very low, right? The lowest level of the library. And what I'm showing you here now is ones that have a limited acute lethality. And you can see again, this region right here is that insecticide region. So these will, even at low doses, will kill them, which is not surprising. It's what you kind of want from these insecticides. But what for us was already kind of interesting or surprising or potentially depressing as you go slightly further is that if you look at long-term lethality, the ones that make it all the way to adulthood, you can see that many of these across the entire library will start to have an impact on how, your overall fitness in this case is how we can judge these, right? So many of these across the library, and some of these are, for instance, fungicides and herbicides, are having an effect on if you'll make it to adulthood, which for us was actually quite jarring already. So now I'm going to walk you through how we actually look at the behavioral shift, right? Because this is something that, again, is proposed for a long time to potentially be augmenting these different populations of insects. So the whole screen was a, kind of a massive endeavor. But I really like this video because it kind of highlights, again, just the complexity of these types of data. So what you can actually do to look at these features that you can start to extract. So I'm not going to go through all of this at scale. There's a huge number of people that I really, really have to thank for this. All of our names are highlighted right here. Because what we've done is basically take all of these and make this large database, this relational database. And what you can see here, these are just kind of nice examples where this is an untreated control where you can see these little wandering larvae. These are types of these phenotypes you can pull out that are slow-moving phenotypes. And many of them will actually do this very fast-moving phenotype, right? This is kind of a classic adversive response in insects, right? So you're not happy, you run. And then if anybody wants this now, I think everything is now live. So, you can go on here and download and play with any of this data right now. So all this is going to be fully available, and I think most of it already is right now. But this is something that for us was quite jarring about the data. And the reason I'm giving these kind of talks right now is that we found extensive evidence for behavioral shifts across this huge amount of this library. In this case, it kind of varies depending on how you will characterize these, but lower estimates, around 45% of these are causing behavioral shifts. And for us, again, what was quite jarring is that you could see that some of these, right? So again, red here, these are ones that are lethal. So, not that interesting in this case. But shown here in blue, these are positive hits now where we're getting very significant effects on the behavior of these larvae, right? And some of these are jarring because some of these are actually, again, fungicides and herbicides, things that classically we wouldn't think were actually that important. So I'm going to show you one kind of correlative evidence or evidence outside of just behavior. Because then we were interested in understanding for, behavior we know is quite noisy, so we want other ways to kind of view this data set. So I'm going to show you here an example of phosphoproteomics, where now we actually went even lower. So these are 0.2 micromolars. These are very, very relevant doses in many cases. And what I'm showing you here is a very classic insecticide, which you could see from the overall proteome, you get a lot of changes, which probably is not surprising as you're dying. Which is coupled now with huge amounts of phosphoproteomic changes across the whole system, right? So you're getting a lot of changes here. We have in some cases, this is actually a fire retardant, which we don't see changes in this case. But now the ones, for instance, these, which is quite active in the media right now for glyphosate, right? This is one that's a herbicide. And here again, we see behavioral shifts. And now we're actually coupling this with phosphoproteomic shifts across these different systems. So clearly they're having an effect on these guys. And then we actually went slightly further to look at the effects potentially if, what happens is that now, we have increasing temperatures happening everywhere. Again, something else proposed to be shifting these systems. So we actually took the 50 most prominent chemicals across these, again, at a very, very low dose, in this case of 0.2 micromolar. And in this case, what we did is we actually looked at now at room temperature, around room temperature, 25 degrees. You can see that, again, these are chosen because they're sublethal. But now if we just heat it up slightly, you can see that you do start getting increased lethality across these. And in some cases, it's actually absolute lethality. So you go from these very slow outs to extreme lethality where everything is dead on the plate. And if you start looking again at the behavioral space, now you can start to see again, if you go from 25 to 29, so this is all normalized data. So you can see that if you're just rolling, so larvae don't normally roll that much. And in this case, you can see now by you pairing the toxin with this increase in temperature, these cases will roll like 16 times more. So basically they're spending most of their time just rolling around a plate, right? So clearly if you're using these in an environment, they're going to have an impact on your system, right? So again, pervasive behavioral changes. So then we want to go even further, and I'm not going to walk you through all the data right now, but all the pairwise effects, but you can also then start to look at the broader scale synergistic effects, right? Because if you go out in the environment and, where this cute butterfly is living, these don't exist in isolation. Typically, these tank mixtures, things that are happening in the environment, are oftentimes different combinations of these different substances that they'd be exposed to. And what I mean by that is that this is an example right here of if we're in Germany. This is actually an example of passive air filtration, and you can see that many of these toxins I'll walk you through right now are broadly manifest around the environment. So we actually took even lower doses, pooled these top 9 toxins present across all of Germany. We exposed them from hatching. And again, just to emphasize, because if you did classic LD50 curves, which is what many regulatory agencies use to allow these to being used very broadly, there's zero toxicity from at least this perspective, from the lethality perspective, right? So again, no lethality across these doses, right? So truly sublethal. But now already from the long-term exposure and compared to that acute exposure I was doing earlier, now we can already start to see that there's getting subtle differences in behavior across these systems. In this case, like larval hunching. Then you can start looking at developmental time. And in this case, already you see it's shifted by an entire day, which for a fly, which your life cycle is about a week, is quite strong. Right, so again, a day. And then for us, for also egg laying rates in this case, now if you look at reproduction rates, this actually is pretty dramatic because these actually drop by 60%, right? So if you're kind of like, obviously you as a more PopGen crowd, These are not subtle changes happening by these chemicals here. Right, so now we're starting to pivot towards, in the interest of time, I don't know how much I'll go into this, but I'll just show you a few cute examples, is we're really going after kind of the larger, broader ecological implication to see how far you can extend these, because it's not just, most people want to kill the fruit flies in your kitchen and most people hate them. So we jumped into something that's a bit cuter. In this case, this is a painted lady, kind of a classic system for developmental biologists in the lab. But the reason we're focused on these, again, there are many species of butterflies. We know they're collapsing. And this is important because these are, pretty major pollinators. So what I'm going to show you now is just kind of an example that kind of, I think, is a very visual way to demonstrate the scale of some of these. So this is the wild type untreated control. These are all different toxins that, again, are sublethal. But I hope you'll see that there are clear differences happening across these. And in this case, you get examples like, let's see, this guy right here, I'll rerun the video. You can see they just spend most of their time just running, right? So much like I showed you those Drosophila earlier, right, which just ran and then exposure to these, we see the same thing across these different insects. Right, and to just kind of slow it down, here's an example of one of these. This particular insecticide is used quite broadly. We know the targets here, but you can see that even at these very low doses, you can see really what they're doing. If you compare them now to wild type and look at the little tracks of those larvae, again, they're spending much of their time just running around these plates. So again, these are used very, very broadly, in this case, across the US, right? So millions of kilos a year. Here's an example of another one. The reason this is kind of an interesting one is you get these very different kind of responses based on dose. And this is a fungicide, right? So again, we didn't expect it to have an effect on the system. And again, this is used quite broadly. So here it looks a bit, well, it's very focal in some regions. In this case, the US, these are still, huge amounts that are being deployed all the time. But not to be all doom and gloom about it is kind of a nice thing, is that there are definitely some insects you want to kill. So a lot of what we're starting to shift into as well is to actually use these from systems to actually look at, in this case, mosquitoes, where we're looking for now, this is just an example of one of these videos where, again, because we can do these behavioral phenotyping, the throughput's super, super high for us. So now we can actually do different libraries, these toxins, to see the effect on the behavior, in this case of the larvae now for mosquitoes, right? So you can track all their videos. This is an example now to extend that same fungicide I had earlier, right? So what I want to emphasize is that much like what we found in melanogaster and the butterflies are not lethal, but you're getting clear effects on the behavior, right? These are just really not moving that much anymore. But the one nice thing is this is what we're looking for right now for some of these screens we continue to go forward, is we're looking for ones that are very, very precisely targeting, certain insects, right? So this precision toxicology approach now to target things you want to kill in a much more, I think, cleaner way, which is hopefully not killing everything else. Because what's also happening right now very actively with a lot of the climate change is, again, you're getting these changes of the expansion of where some of these are live, right? So we have, in Germany, we're starting to get some of these different mosquitoes. This is actually old because more people have caught malaria recently in the US, right? So you're getting this big expansion now as we have all the climate kind of shifting, right? So again, we think we need much more sensitive assays as we approach these from a chemical perspective. But again, kind of talking now to, a population genetics group, we think that these type of approaches right now, particularly from the behaviors, if anybody has any interest in the behavior, we're like all in right now because we think this is, the volume of data we can process is massive. Right, so now we're starting to look at different selection pressures, again, agricultural toxins, pair it with things like temperatures if we need to, doing different developmental screens. And again, a lot of what we're starting to do now is do, hypermutate different networks. And then again, to compare this now to different natural isolates, right, to kind of this feed back and forth for what we're doing in the lab and trying to connect it now with a more meaningful way of things we're seeing in nature. And again, for a lot of us to go after these kind of classic evolutionary questions about dynamics, predictability, kind of things many of us here are interested in understanding. So I think at this point, I'll kind of stop. I think I hit my time fairly reasonably, which is good for me. I tend to go on and on. So thank you guys very much for an audience. And again, now, we're having major turnover of the group. All these people nicely left, got a good position. So if anybody's ever interested in reaching out about possible, working with us, we'd be more than happy to discuss. And I'll stop, and if anybody has any questions, I'd be happy to address. So thanks.